ASAP has the capability to model fluorescence in optical systems. Coupling this capability with scatter and absorption in tissue phantoms provides a useful feature set to help examine bio-optical devices such as fluorescence microscopes. In this example, we've used the realistic skin model to create a tissue phantom, and then placed within that tissue phantom a fluorescing body, which we can excite and then use the energy from to create a new source for re-emission. And what we see is a skin model composed of the stratum corneum, the epidermis, and a dermal layer. The dermal layer includes our fluorescing body. The blue rays that you see here are actually our excitation rays, and that excitation is around 515 nanometers, which is the excitation for yellow fluorescent protein, which we've modeled here. The yellow rays that you see are actually the re-emission rays from the yellow fluorescent protein, and as you can see, they've scattered within the volume. So we're actually modeling the real effects of scatter and absorption of the dermal, epidermal, and stratum corneum layers, so that when you take a look at the distribution of light as it comes out of the system, you can compare that with the volume that you've created within the system to see how it differs as far as size and shape. And as we can see, there's a very marked difference in the size of the distribution of energy on our detector versus the size of this particular yellow fluorescent protein body. Now let's take a look at how we actually accomplished this. First, let's run this file and take a look at how we deposit energy within this skin. Now what you see in the blue here is the excitation source at 515 nanometers, and the yellow sphere within the dermal layer is our yellow fluorescent protein body. Now we're depositing energy in such a way that we can track the energy within that volume and then use that energy to create a new source. Basically what we'll do is turn the source inside out and re-emit the source, and the energy is going to be scaled based on the quantum efficiency, so how well the fluorophore can actually re-emit that energy, which in this case is about 61% for yellow fluorescent protein. And as we see here, we have a distribution on a detector that we've created just above the surface of the skin, and it's a very, very diffuse sort of energy distribution, which is what we would expect from scattering and absorption from re-emission of a source within another scatterer and absorber. What we can do with this is we can actually use this to examine optical systems that would work with the skin, such as fluorescence microscopes, so that we can optimize or idealize our optical system to get the best image quality out. So to summarize, what we've accomplished is creating an excitation source which has deposited energy within a yellow fluorescent protein body. We have taken that energy and re-emitted it as fluorescence. And the fluorescence is scattered and absorbed within the skin in a realistic manner, and we have collected on the surface of our sample the energy as a distribution that can be used in a very realistic manner to examine fluorescence systems in skin tissue.